participants in Europe, and good morning to Professor Schwartz and others logging in from the US. I am Sankalp, faculty in the area of innovation and entrepreneurship in the School of Management and Entrepreneurship, and associate faculty in the AED school at IIT Jodhpur. Let me begin by thanking you for your presence, and let me take a few minutes for preliminary introductions. Indian Institute of Technology Jodhpur was established in 2008 to foster technology education and research in India. The institute is committed to technological thought and action to benefit the economic development of India, scholarship in teaching and learning, scholarship in research and creative accomplishments, and relevance to industry are three driving forces for us at IIT Jodhpur. Let me now introduce to you the panelists for the day. Dr. Shantanu Chaudhary is the director of IIT Jodhpur. He holds a BTech and a PhD from the Indian Institute of Technology, Kharagpur. Professor Chaudhary is a fellow of Indian National Academy of Engineers and National Academy of Sciences. He is a fellow of International Association Pattern Recognition, and he was awarded the Indian National Science Academy Medal for Young Scientist in 1993. He received ACCS CDAC Award for his research contribution in 2012. Professor Chaudhary has about 300 publications in peer-reviewed journals and conference proceedings, 15 patents, and 4 authored books to his credit. Professor Chaudhary is a recipient of the Distinguished Alumnus Award from his alma mater, IIT Kharagpur. Dr. Sampat Raj Vadera is professor and head of the Department Physics at IIT Jodhpur. With a PhD in physics from Jai Narani University, he was the director at the Defense Lab of Defense Research and Development Organization, and he comes with over 35 years of experience of translational R&D before joining IIT Jodhpur. His area of expertise lie in solid-state physics, material science, nanoscience, and nanotechnology, stealth materials, and stealth technology. Dr. Neeraj Jain, with a PhD from Indian Agricultural Research Institute, is the former director of National Brain Research Center, an autonomous institute of Department of Biotechnology, Ministry of Science and Technology, Government of India. He serves IIT Jodhpur as professor in our Bioscience and Bioengineering department. His area of interests are around neuroscience, mammalian sensory and motor systems, tactile information processing, brain plasticity, and brain-computer interface. We have Dr. G.S. Toteja, Chief Executive Officer of Jodhpur City Knowledge and Innovation Foundation, which has been established under the Principal Scientific Advisor, Government of India, with IIT Jodhpur as the nodal agency. Dr. Toteja was formerly Additional Director General at the Indian Council of Medical Research, New Delhi. He was a director, ICMR, National Institute for Implementation Research on Non-Communicable Diseases, and he was the head division of nutrition at ICMR, New Delhi. His area of interests are around nutrition research, food safety, and health research. Dr. Mayank Vatsa is a professor in Computer Science and Engineering Department. He received his master's and PhD in Computer Science from the Lane Department of Computer Science and Electrical Engineering at West Virginia University. He was a member of the Indian Biometric Standards Committee for e-government applications. He has also served as a member of the UIDAI's Biometric Standards Subcommittee. His area of interests are around biometrics, image processing, machine learning, deep learning, image forensics, and information fusion. Dr. Richa Singh has received her PhD degree in computer science from West Virginia University. Her area of interests are pattern recognition, machine learning, and biometrics. She is a fellow of IAPR and a senior member of IEEE and ACM. Dr. Rajendra Nagar has a PhD in electrical engineering from IIT Gandhinagar. His area of interests are computer vision, image processing, computer graphics, and geometry processing. Dr. Manish Narwaria has a PhD in computer science from NTU Singapore. His area of interests are around multimedia signal processing. Dr. Ranju Mohan has a PhD from the Indian Institute of Technology, Madras, and a postdoctoral fellow from Indian Institute of Science, Bangalore. She is the head of the Department of Civil and Infrastructure Engineering. Her area of interests are around traffic flow theory, macroscopic and microscopic modeling of traffic flow, connected and autonomous vehicles, and dynamic traffic assignment. I am Sankalp Pratap. I have an MBA and PhD from Indian Institute of Management, Calcutta. I am an associate professor in the School of Management. My areas of interest are around healthcare entrepreneurship, indigenous entrepreneurship, and art entrepreneurship. I now request Dr. Mayank Vatsa to introduce the AI School and the Thar Talk series. Dr. Vatsa, please.
Thank you, Dr. Sankalp. IIT Jodhpur established in December 2020 the AI and Data Science School, or we call it as AID School, which was inaugurated by Professor Jeffrey Hinton, who is a Turing Award winner and widely considered as a father of deep learning. The school actually plans to promote cutting-edge technology education and research with the ambition of this young school is to establish itself at the forefront of AI research and to become a globally coveted destination for technology development, entrepreneurship, and education. So all three pillars of technology development, research, entrepreneurship, and education is the foundation of this particular school. So while we have been engaging conversation inside our school with the introduction of our Thar Talk series, we have been stepping out to converse, engage, and exchange ideas with global experts and local enthusiasts. And in this regard, today we are delighted to introduce you to our Thar Talk series, which is conducted by AID School in collaboration with Jodhpur City Knowledge and Innovation Foundation. The Thar Talk series will focus on the multidisciplinary emerging themes. It will address the fundamental issues and explore path-breaking possibilities in various disciplines of engineering and sciences. It will encompass several fields of interest in the AID School, including brain science, public healthcare, economics and entrepreneurship, robotics, education, public ethics, along with the fundamentals in AI and data science. With this, I would like to invite Professor Neeraj Jain to introduce our today's keynote speaker. Professor Jain, over to you.
Thank you. Thank you, Professor Mayank. It's my great pleasure to have Professor Andrew Schwartz as the first speaker in this series of talks. Yesterday was the National Technology Day, and what you will see in his talk is, I would say, the peak what technology can achieve, how a paralytic person can control a robotic arm with such a fine degree of precision. Professor Schwartz obtained his PhD in physiology from University of Minnesota, and then he did his postdoc fellowship at Johns Hopkins School of Medicine with Dr. Georgopoulos. And that was the time when they discovered that you can actually predict which way arm will move by recording from a number of neurons or a single neuron, and repeatedly you can calculate a vector, and that vector direction will give you the direction in which the arm will move. So essentially that was the beginning of a population coding in its full glory. His Georgopoulos work and his work at that time excited the field as much as I can say Yuval and Wiesel work on neuroscience and in somatosensory cortex at that time all the work that was coming out, which was telling us how the brain works, which was done by Mountcastle. So after he did work at Burroughs Neurological Institute in Arizona, and then Neuroscience Institute in San Diego, and then he moved to University of Pittsburgh in 2002, where he is a distinguished professor of neurobiology and holds an endowed chair in systems neuroscience. His work is amazing in the sense that one of the few labs in the world who have actually implanted an electrode array in the brain in patients and showed that how they can control the robotic arm, as I said, or do the very complicated task. I invite Professor Schwartz for this talk. Thank you, Professor Schwartz, for being with us today.
Thank you, Dr. Jain, and thank you very much for inviting me. Let me get started with the talk here. All right. So I like to give these talks in a way that shows the engineering advances, but what I really want to emphasize is the underlying science that makes the engineering possible. So I like to start out with this particular picture to show you the nature of the problem. And I'm interested in movement, volitional movement, in particular arm movements, and what the relationship is between neural activity in the brain and these arm movements. So if you just take a step back, and I illustrated it with this cartoon, and look at the problem. The problem is that you have something like 10 to 20 billion neurons in the cortex, and almost all of them are active almost all the time. And so if you had a magic machine where you could actually look at these neurons and see when they're active during a task, this is what you would see. You'd see billions of neurons throughout the entire brain firing together. And there wouldn't be any obvious relationship between one neuron and another. And it would be relatively noisy. It would be a difficult task to just look at a picture like this and try to gain an understanding of what's happening. But that's really what our task is. Our task is to learn something about the structure of firing in these neural populations. So let's just look at the basic element that we're going to be studying, and those are single neurons. You may remember this from your basic biology course. A neuron has a cell body, it has dendrites, and it has an axon, and it gets its input from other neurons through synapses. Typically in the cortex, there are 10,000 synapses on every neuron. And in order to generate an action potential, which is a pulse of electricity that travels down the neuron from the cell body to its axonal terminals, you need to have a collective action of these synaptic inputs. So every time one of these synapses is active, it generates a little change in the voltage of the membrane and the dendrites. And if enough of these excitatory impulses occur simultaneously, the neuron will reach a threshold. So this is a nonlinear event. And when that threshold is reached, an action potential is generated that travels down and communicates with the neurons that are connected to the axon terminals. And so here's the problem. We have 10 to the 12 neurons in the cerebral cortex. Each neuron has 10,000 dendritic terminals per neuron. And if you want to try to understand what's generating a pattern of activity, you need to consider 10 to the 15th possible input states at every instant. So you can see this is a tough nut to crack. This is a difficult problem. And I think it's important to remember that this is really the key piece of knowledge that we need to have in order to begin to understand brain function. Okay, so what are we going to do? When an action potential is fired, it generates an electric field. We're going to put a microelectrode near the neuron so we can intercept that electric field and actually see these action potentials as they occur, these electrical events. We're going to have a monkey do the same task that I showed you in that puppet cartoon. The monkey's going to draw a circle with its arm. And while the monkey's drawing the circle, we're going to record with microelectrodes, single neurons in the monkey's cerebral cortex, in a particular part of the cortex called the motor cortex. So you'll see when I start the movie, you'll see the monkey's arms start to move in a circle. Here's an oscilloscope readout of those action potentials. An action potential is about a millisecond long. The waveform is consistent, so we can see that these action potentials are coming from one individual neuron. And what I want you to try to understand is the relationship between the firing rate of these neurons and the way the monkey moves its arm. So you can see it's highly modulated. When the monkey rests, there's a background activity. When he moves up, there's a burst of activity. When he moves down, it shuts off. So you can see there's this very consistent relationship between the direction that the arm moves and the firing rate of this neuron. Now, this is a demonstration of the relationship between the direction of arm movement and neuronal firing. And this was a basic observation made by Apostolos Georgopoulos in the early 80s. And what he had the monkeys do was to move from the center of a tabletop, whoops, let's go back, from the center of a tabletop to one of 8 targets arranged in a circle around this center position. So the monkey would start by capturing a center light that would be extinguished, and one of these 8 lights would be lit, and the monkey had to move this draftsman arm over the tabletop to one of these 8 targets. This is what we call the center out task. And what it was doing was looking at the parameter of movement direction. So while the monkey performed this task, a single neuron was recorded in the motor cortex. And every time the neuron fired an action potential, we put a tick mark down in one of these rasters. So here you see the monkey was moving straight up on the tabletop, so from here to here. And every time, this is time along here, every time the neuron fired, a little tick mark would be put down on this trace right here. And these traces were aligned so on movement onset. So this is where the movement began. And each movement was repeated 5 times. So you can see a pattern in the raster. And what you see here is that shortly before movement onset, the neuron started to fire at a high rate. So this was movements going upwards. And when the monkey went in the opposite direction from here to here, you can see that that neuron stopped firing before the movement. And in between was a gradual change in this pattern. So as he moved in directions farther away from the straight up, the neuron fired less and less. So what we did was summarize this firing rate. What we did was average across the 5 repetitions and then across time to get a single mean firing rate. So in this case, we got a firing rate, so number of action potentials per second, okay? And this is represented by a single number. In this case, this is 90 degrees, so this would be the firing rate for movements upward. And we did this for each movement, plotted them on movement direction here, and you can see that we get this pattern of points. And what we did then is fit this pattern with a cosine function. So the cosine function has a period of 360 degrees. And the phase of this is determined by the direction that the neuron fired fastest. And we call this the preferred direction. So in this case, the preferred direction is somewhere around here. Okay. So what we're doing essentially when we fit this cosine function is we're applying a model to our data. And I want to take a little bit of time to just talk about that model. So here's our model written out as an equation. We have firing rate on the left. We have movement direction over here. This is a vector, an XY vector on the plane. And these are the coefficients of those factors. And you can see that this is an equation for a plane. If you took away this by, my, you would have an equation for a line. So this is an equation for a plane. It's the simplest possible equation you can have to describe this type of data. Okay, so we can rewrite this in terms of vectors. So we have a vector for our slopes, our regression coefficients, and we have a vector for our movement direction. And we can express that as a dot product. So again, these are all the same equations. I'm sure you're familiar with this. We can rewrite this dot product as the cosine between the two vectors times their magnitudes. And to just give you a graphical representation of this, suppose I have a movement vector up and to the right. So a target was lit, the monkey starts down here, a target was lit up here, the monkey moves from here to here. We have a movement vector in this direction. And we have a preferred direction, like we did in the last cell, that was basically straight up. We project this vector onto this B vector. The magnitude of this B vector is the maximum firing rate of the neuron. And since we're moving at an angle slightly away from that preferred direction, the firing rate is slightly lower than its maximum. And you can see as you move in different directions, here the firing rate gets very small. This neuron would fire at its background firing rate. If you move opposite the preferred direction, the neuron starts to shut down. But the important thing to remember here is that our model is just a projection of two vectors, and the firing rate is determined by the cosine of the angle between these two vectors. So this is, again, a very simplistic model, but it's powerful in that we can arbitrarily add terms to this equation and the form of the model stays the same. All we do by adding extra terms is increase the dimensionality of these vectors. It's still the angle between the vectors that determines firing rate, independent of the number of dimensions of each vector. So you can think of this as a geometric representation of correlation. The more the combination or the ratio of parameters in the movement vector matches those that make the neuron fire its fastest, the faster the neuron will fire. So that's the basic concept. So what I just described is what we call an encoding function. We have firing rate on the left side of the equation and movement parameters on the right side. So we're basically saying, what's the combination of parameters that makes a neuron fire? We can invert that by saying, given a firing rate of a neuron, what were the parameters that made that neuron fire? So that's what we call decoding. And in order to do that, we have to look at a population of neurons because these cosine functions are rather broad and noisy. So what we found is that many neurons in the motor cortex fire in this manner simultaneously. Each has a different preferred direction. So we represent this population by a blue vector, and the vector is in each of these neurons' preferred direction. So in this case, we have a collection of about 300 neurons. So the direction of each blue vector is the neuron's preferred direction, and the length of the vector is how fast the neuron's firing at a particular point in time. So we take a snapshot of the population, make this cluster, and then we just take a vector sum of all these contributing vectors and get a population vector. And the population vector is our readout of the population and predicts the instantaneous movement direction and, as it turns out, the speed of the movement at a particular point in time. So if we have the monkey perform a drawing movement, like drawing in a circle or drawing some other arbitrary shape, we can take a series of these snapshots through the movement, look at the neural activity, and predict the instantaneous velocity of the hand. So I'm going to show you the results of this. These are real data now. And what you can see that is, if you do that and add these population vectors tip to tail, you can recover the trajectory that the monkeys drew. So in this case, the monkeys were drawing spirals from outside in and inside out. And you can see this technique gives us an isomorphic representation of the instantaneous speed and direction of the hand. All right, so this is our basic mechanism that we're going to use here. Now, what I just showed you was a trajectory. And if you step back a second and think about it, trajectories are equivalent to behavior output. What do I mean by that? Well, the way we express our behavior is by moving. And what I mean more explicitly is that the only way you know about what I'm thinking about, what's occurring in my brain, is by the fact that I move. So you can think of movement really, instead of the physics of energy and muscle contraction and displacement, you can think about it as a communication channel. It's a communication between the covert operations in my brain and the explicit outside world. So if you lose the ability to move, you lose the ability to communicate. All right. So that's the genesis for neuroprosthetics. We want to give paralyzed people the ability to communicate and to move again. So in order to do that, we need to record from a population of neurons simultaneously. That technology has been developed in the last 20 or 30 years. What we're using now is a relatively old technology. It's called the Utah probe. It's a platform built out of a solid piece of silicon. It's etched and carved to form an array of 10 by 10 electrons, or 10 by 10 electrodes. Each electrode is 1.5 millimeters long, and it has a platinum iridium recording tip. We turn this upside down, inject them into the brain. So here's the motor cortex of a monkey. Two of these arrays are 2 by 2 millimeters in dimension, are in the motor cortex. This is the hand and wrist area of the motor cortex. This is the shoulder and elbow area. So we can record from 100 electrodes here and 100 electrodes there to get our population that we need in order to decode the intended movement. So here's the paradigm. We take these Utah arrays, put them in the motor cortex, record from individual neurons, find each neuron's preferred direction, add all these neurons together to get a population vector. That population vector gives us an instantaneous velocity. That velocity then is fed into a robot controller, and the robot controller will move a robot arm in that velocity dictated by the population vector. So in this example, we now increase the dimensionality of our model. So we added a Z dimension, so the arm can now move in free space, three-dimensional space, and the monkey can control a gripper at the end of the arm. So now we have 4 dimensions, but as I said, it's the same basic model. There's 2 vectors, a movement vector and a neural vector. So here's a demonstration showing how this works. We can put marshmallows out in front of the monkey. He can operate this arm, close the gripper, and feed himself. And we can put these marshmallows any place in space, and he's able to operate this arm with his neural signal. Okay, so that validates the model. What we did is added more dimensions to the model. So now we added a wrist that can move in 3 dimensions, yaw, pitch, and roll. And then now the monkey has to orient the hand to the cylinder in order to get a reward. So again, you can see the monkey's capable of doing that. Here's our model. You can now see that it's 7-dimensional. We have X, Y, and Z displacement, yaw, pitch, and roll of the wrist, and closing the aperture. So now we're up to 7 dimensions. So the point here is that these neurons don't just encode one thing, right? We're showing that they encode at least 7 things simultaneously. Okay, so we wanted to expand this so that we can now decode fingers as well as the arm. So we had the monkey do a task in which he reached a variety of different objects. And in order to do this, he had to change his hand shape. And we measured hand shape and fed it into a dimensionality reduction algorithm so that we have 4 fundamental hand postures. And we put those 4 postures into our model. And you can see here that we have now 10 dimensions. So again, we have X, Y, and Z displacement, yaw, pitch, and roll of the wrist, and these 4 principal components that describe hand shape. So here's the monkey hand shape, his actual hand shape compared to what we calculated from the population vector as he reached for these objects. So here's his actual hand shape. Here's what we calculated. And you can see that we're doing a very good job. Okay, so again, what we've done with this demonstration is validate the model. So we started working with the US government, the defense agency, DARPA. They had a project that was $150 million. Most of it went to developing this very elegant robotic prosthetic arm and hand that was capable of most of the natural degrees of freedom of a real arm. And here you can see at the Applied Physics Lab where they developed this, we have a demonstration with a data glove that measures finger and wrist. And you can see as the human subject moves his hand, this artificial MPL, it's called modular prosthetic limb, mimics it very nicely. Okay. So at this point, this was about 2011, 2012. We thought we had everything we needed to start human trials. We showed that our algorithm worked, that we could record from many neurons simultaneously, and that we had a device that was capable of replicating natural arm movement. So I'm going to play this little clip here that sort of tells the story, and then go on from there.
In a decade of war, more than 1,300 Americans have lost limbs on the battlefield. That fact led the Department of Defense to start a crash program to help veterans and civilians by creating an artificial arm and hand that are amazingly human. But that's not the breakthrough. We don't use that word very often because it's overused. But when you see how they have connected this robotic limb to a human brain, you will understand why we made the exception. Andy Schwartz, on the right, is working on an ambitious Defense Department project called Revolutionizing Prosthetics. 4 years ago, we visited his lab, and Schwartz showed us how he implanted tiny sensors like this one into the brains of monkeys, and then wired them to a crude robotic arm. Schwartz told us that when the monkey thinks about moving his own arm, his brain cells or neurons fire off electrical signals. The sensor in his brain can pick up these signals and send them to the robot. So he's operating the arm in 3 dimensions, up, down, forward, and back, as well as the gripper. What you're telling me is that the monkey is operating this arm with nothing but his thoughts. Absolutely. What are the chances that a human being would be able to do the same thing? Oh, we think a human being could do much better. That conversation was in 2008, and since then, the $150 million Revolutionizing Prosthetics program has reached farther than most thought possible. Awesome. Dr. Jeffrey Lane, a retired Army colonel and neurologist, is in charge. After seeing the wounded on several tours in Iraq and Afghanistan, he told his team that he wanted a breakthrough within 5 years. Did any of them say, look, Colonel, we're not sure we can do this?
Oh, absolutely. They thought that we were crazy. But that's quite all right because I think it's in our insanity that things happen.
Who wouldn't want to do this? When they told me I heard about the study, I said, oh, absolutely. I couldn't not do this.
Last February, Jan Sherman put herself on the line for a more sophisticated version of the surgery that they had done earlier in the monkeys. The procedure was done by University of Pittsburgh neurosurgeon Elizabeth Tyler Cabarro. During the 6-hour surgery, 2 sensor arrays, each the size of a pea, were placed on the surface of Jan's brain. They plugged her brain into the computer, and this is what we saw.
I can move it up and straight down and left and right and diagonally. I can close it and open it. And I can go forward and back.
That is just the most astounding thing I've ever seen. Can we shake hands?
Sure.
No, really.
Yeah.
Like, come right over here?
Yes, you come over there.
Okay.
Rest your hand. There we go.
Oh my goodness. Wow.
And I can do fist bump if you'd like.
That's amazing. What are you doing, Jan? What's going on in your mind as you're moving this arm around? What are you thinking?
Okay, the best way to explain it is raise your arm.
Uh-huh.
Good. Now,
Right.
what did you think about me did that?
Well, not very much. I do it all the time.
Exactly. It's automatic.
Is that hard work? Are you having to concentrate?
No, it was hard work getting there. I struggled greatly to go up and down, getting now up and down. So we say, I don't even think about it, side to side. Don't even think about it.
Just like
Just like that.
your arms used to.
Yes.
Okay, so the point here that, that I really want to stress is that this is a natural kind of movement. She's not thinking about wiggling her little toe to make the arm move to the right or left. She's thinking much like you and I, when we want to move our arm, it's more or less very natural kind of thing to do. And I want to just play this one clip exercise that we had her do in the lab. And I watched the movement of the arm. And I think it's very convincing that this is a very natural kind of movement. Here's Jan down here in her wheelchair. Look how graceful and coordinated and smooth these movements are. Okay, so to me, this is one of the most convincing parts of the demonstration. Okay, so Jan was our first subject. We had a second subject, Nathan. And in addition to putting electrodes in his motor cortex right here, we put electrodes in his sensory cortex as well. These electrodes were for stimulating. And the idea was that if we stimulated with small electrical shocks in his sensory cortex, he would register those as sensations coming from his finger. So we had this prosthetic hand. It had pressure sensors built into it, torque sensors in the joints. And the idea is that once these fingers encountered an object, these sensors would be activated. We would translate that activation into pulses that were sent to a sensory cortex, and he should register that as touch. So here's a demonstration. This is Rob Gott, who is leading this part of the study. This is Nathan, he's blindfolded. Rob's going to push on these fingers. Pinky, index, pinky, right. Okay, so you can see that this worked. And then I just want to show you President Obama, while he was still president, came out to Pittsburgh and actually met Nathan. Again, this is Rob Gott, who led this along with John Collinger, another professor here at Pitt.
They implanted 4 microelectrode arrays into his brain, each about the size of half a micron. Those implants connect neurons in his brain with a robotic arm. So today he can move that arm the same way you and I do, just by thinking about it. Mason is also the first person in human history who can feel with his prosthetic fingers. Think about this. He hasn't been able to use his arms or legs for over a decade. Now he can once again feel the touch of another person. So we shook hands. He had a strong grip, but we kind of toned it down. And then we gave each other a fist bump.
Okay, so I think this tells you, gives you some demonstration, and to me it validates some of our assumptions, some of our models, showing that these neurons encode direction very robustly, and that by using the idea of population decoding, we can recover a lot of the information that's present during these natural tasks. Now, what I want to do is dive a little deeper into the science of this kind of behavior and show you some of our more recent work. So let's again start with this basic center out task. And what I want to do is, explore some of the assumptions that we made initially. So remember the way we did this. We took the mean firing rate, and this was our metric for describing the behavior of these neurons. And so essentially you take this whole pattern and just express it with a single number, the mean firing rate. Now, here's the problem. If you actually look at these patterns and make histograms out of these, you see that there's not just a mean constant firing rate, but that there's a time varying pattern. And it turns out that this time varying pattern is important. It gives us some more clues as to what's going on functionally with this neural activity. So let's look at our assumptions. So let's say we move in 8 different directions, and we have color-coded firing rates down here. And this axis is time, this axis is firing rate. And our initial assumption was that at the beginning of movement, the neuron would turn on, fire at a constant rate, and then turn off at the end of movement. And that these firing rate intensities were cosine tuned so that they had a pattern that looked like this. Now, if we looked through time and took time slices through these, plotted these points, made a cosine function, and calculated the preferred direction over time, when you have a pattern like this, you'd see that the preferred direction was constant across time. So this idea of a single preferred direction as the task takes place, this stationarity assumption, would hold in this case. Now we know that neurons don't fire this way. They fire in time-varying patterns. And so we have a pattern that looks like this. So the profile is just scaled in time, but the profile remains constant. And we take these slices through, we see that our stationarity assumption is still valid. Okay, if we were to calculate preferred directions across these different points in time, we would see a constant preferred direction. Where our assumption fails is that the profile changes during time. So let's just look at this bump here. This would correspond to movement straight up. So we move straight up, but later in time, if you look at the other directions of movement, you see that this guy is no longer the winner, right? In fact, as he goes through at the end, it fires at the end of the movement at the least amount when you move in that direction. So if you were to go through and calculate preferred directions in time, you see that there would be a change of preferred direction. And this would now violate our initial assumption. So we need to look at this idea in more detail. If we take a real neuron, and now the monkey was moving in 16 different directions, so these are color-coded directions, and we look at a single trial, and we take this idea of calculating cosine functions at different points, we see that if we calculate a cosine function here and take these 16 points and plot them down here, we get a cosine function with a preferred direction of minus 90 degrees. And you can see that if we do this throughout here, we get a pretty constant, consistent tuning function until we reach this point here, where there's another bump of modulation. And if we take the firing rates at this point, calculate a cosine function, right? We get a completely different preferred direction. In fact, it switched 180 degrees. So now the preferred direction is plus 90 degrees. It's constant for a while. And then all of a sudden, there's a jump over here at the third modulation. And you can see that we're back down to minus 90 degrees. Okay, so what we see are these clear epochs, in this case 3, in which we get these different bursts of firing. And each burst, each epoch seems to be associated with a different preferred direction. So why is this important? Well, imagine that we're thinking about this like an engineer, and we have a simple linear systems, And we have some input coming into our system. In this case, the input is movement direction. And when you're reaching straight from a center to a target, the direction is constant. So you can think of this as a constant input. And then out here is the firing rate. And this firing rate will obey some kind of cosine function. And the cosine function is generated by a transfer function in this black box. And so what seems to be happening is you get the same input and different outputs depending on where you are in the task. So this makes the engineering problem very difficult. We have a complex system that is not conforming to our usual engineering principles. So let's look at a variety of neural data here. So these are examples from 2 different monkeys. Here we get a firing rate, again, a burst of modulation. This is time. This is where the target's presented. This is where the monkey starts to move. Here's maximum movement. Here's the end of movement. So right here, we get a burst of activity before the onset of movement. And we can calculate cosine functions through this. And these preferred directions are constant throughout the task. Here's an example where we had 2 bursts of activity. And if we calculate preferred directions here, we can see that we get a preferred direction for this burst right around movement onset, and another one over here that seems to be associated with the ongoing movement itself. Here's another example for another neuron. It again has 2 bursts, and this step-like function or step-like change in preferred direction. Here's a neuron, like I showed you before, that has 3 bursts, and again, 3 distinct stages of neural firing. All right, how do we think about this? One thing that we found is that these peaks seem to occur in the same place for all neurons. So there seems to be this system-wide set of 3 epochs, so that if you get a modulation peak, it seems to occur at the same point in time. So here are the peaks for 68 units in one monkey, and you can see that they tend to occur at the same points in time. Around movement onset, somewhere in the middle of the movement where maximum speed is, and then at the end of the movement when the monkey's acquiring the target. Okay, so there seems to be this global set of 3 neuronal states. Somehow these neurons are being modulated. They change their preferred directions in a consistent way throughout the task. Inconsistent meaning they change. They don't change. The shift in preferred direction is more or less random. We haven't seen any kind of structured amount in the amount of rotation or the amount of change in preferred direction. So we're going to make a model of this, and we're going to model these epochs as 3 distinct bursts, one that occurs early, one that occurs in the middle of the movement, and one that occurs late. So we have this template of 3 Gaussians, and what we're going to do is assign preferred directions to each of these, and we're going to randomly assign preferred directions to each of these. And then we'll say the monkey's moving in 16 different directions, and we'll get the modulation of the activity for each movement and add these components together. So here's one example where we randomly chose these preferred directions, went through 16 different movements, added these scaled templates together, and got these firing rate profiles. Okay. So what we found were 3 bumps. They're used as a template to simulate firing rates during a center out task. Each bump was cosine tuned to direction with a randomly assigned preferred direction. Since the modulation of each epoch was randomly assigned, we might assume that there would be no correlation between epochs or between the firing rates of different simulated neurons. So we want to test that, okay? And we're going to test that by using principal components analysis to look for correlational structure. Okay, so we calculate an n-by-n correlation matrix using the firing rates of all the trials and all the targets, and we calculate principal components from this data matrix. And this is what we got from our simulation. So what I'm showing you are the scores. So these are the data projected onto the eigenvectors. And we found that 6 of these components accounted for more than 90% of the total variance in the population. And you can see that each of these components, these are color-coded for movement direction. Each of these is cosine tuned, okay? And that these seem to occur in pairs. So you have a pair that occurs early in the movement, in the middle of the movement, and the end of the movement. And if you look at these profiles, they match the template. So essentially what's happened here is our principal components have recovered the structure that we use to generate the simulated activity, okay? And the only way we can do that is by finding very clear correlational structure. So somehow, even though we generated these firing rates with random epochs, we are able to recover correlational structure. Okay, so let's look at real activity. So we recorded activity from 80 some odd cells in a monkey simultaneously. And voila, these are actual scores. And you can see they perfectly match the simulated data. So they're cosine tuned. They occur where those templates occurred. And it works very well. Okay, so it says that a sequence of 3 directional signals drive the population for each reach. The cosine tuning of direction is reflected in the population activity and is responsible for the correlational structure detected by principal components analysis. Remember, the only thing that we did was to say that these fire rates have to be cosine tuned in each epoch, okay? And that was enough to generate very strong correlational structure. Notice that in the simulation, the fire rates cannot be deterministic since the sequence of epochs were chosen randomly. So I want to talk about what I mean by determinism. If we take a standard equation for our dynamical system and we look at it applied to neural activity, we say that the change in firing rate, so this would be a vector of neurons, different neurons, and we take this and we look at the change in this matrix over time. We say it's due to some linear function of the current firing rate plus an input. Okay, so this is the idea of a deterministic system, that something at one point in time determines what happens later, okay, or the change in activity. So if we say that, right, so the important thing is that people applying this to the nervous system take the idea now that U of t is 0, so that there's no input. What they're saying is that everything that's happening is due to intrinsic processing. So in our case, the firing rate patterns that we see in the motor cortex are due to neuron-neuron interaction within the motor cortex. So the motor cortex is isolated and generating these intrinsic patterns. Okay, where does this idea come from? So this is work that was done by Churchland and Chenoy and their colleagues. They used a delay task, which was a center-out task. Target would come on. Instead of the monkey moving as soon as possible, he was told to withhold his movement until a goal cue occurred, and then the movement would occur. This is called the delay epoch. What was assumed is that there was some sort of preparatory activity taking place during this delay that dictated what happened later in the movement. So what they did was they formed a regression, and they said, we can predict what happens earlier in the movement by looking at the principal component scores, the population activity that happened later, okay? And if our regression is valid, we should be able to take the predicted firing rate coming from this equation, match it to actual firing rate, and get a nice linear relationship, okay? And in fact, when they did that, they did find this linear relationship. Okay. They also assumed that the system was oscillating. Why did they make that assumption? If you take 2 time-varying patterns plotted on separate axes and see a circular trajectory, you could take this as a sign of an oscillatory process. Schwartz and colleagues called this rotational dynamics. And what they saw is that they took one of their principal component scores and plotted one against another one. Over time, they would see this rotational pattern. Okay, they call this rotational dynamics. And when they saw this, they said, oh, the system must be oscillating, because when you plot an oscillating system on a phase plane like this, you see this rotation. So let's just look at this. If you take sinusoids, time-varying sinusoids, put them into an XY scope, so you put this on the X scope, this time scale, put this on the voltage, and plot them against each other, what you see is a nice perfect correlation when they're in phase. You see the circular trajectory when they're out of phase. If they're 90 degrees out of phase, you get a perfect circle, right? And then you get different slopes depending on the phase relationship between them. So this is sort of the cardinal sign of an oscillating system. And this is what they thought they had, okay? All right, so what do we see in our simulated data? So in our simulated data, remember, there is no determinism. So we take the same equation, we look at our data, we take the firing rates in the pre-movement period, and we plot them against the scores that we got later in the movement. So we take neural activity early in the period, and we use these scores out here in our regression equation. We plot them out, and in our simulated data, we get almost perfect correlation. In our actual data, we get very good correlation. Okay, so in our data with no determinism, we still see this relationship. Notice now when we plot our components against each other, we get this circular pattern. So if we plot, in this case, score 2 against score 6, right, we get rotational dynamics, and we get the same thing in our real data. If we plot components of the same phase, we can recover the metrics of the movement very nicely, both in the simulated data and the actual data. So how can we get the same results as Churchland without determinism? So let's go back to our cosine function. Now think of these cosines as our tuning functions. And notice that when they're in phase and we plot one cosine against another, and they have the same period, the same frequency, we get perfect correlation. Think of this as correlated activity. Right? Even if they're slightly out of phase, we still get very good correlation. Only when they're 90 degrees out of phase do we get zero correlation. Okay, so just by having cosine relationships, two different functions, they have the same period. If they're cosine tuned, they're going to have correlated activity. So if we look at our equation, these scores are cosine tuned, the firing rates are cosine tuned. So of course, we're going to have a good regression. All right. So let's go back to these bumps. How is it that when we plot these bumps, we get rotational dynamics? Well, let's look at this. We have two bumps, say from two components. When they're in phase, again, you get perfect correlation. But look at what happens if they overlap, right? You start getting these circular patterns, rotational dynamics. So there's no oscillation here. We just have two separate bumps, two discrete bumps. And as long as they overlap, you get rotational dynamics. All right. So to conclude, there's no evidence of oscillatory neuronal processing. Rotational dynamics just comes from overlapping bumps. Cosine tuning in a population of neurons can generate correlational structure. During a singular reach, there appears to be 3 states taking place. And these states are likely due to latent drive in the form of directional information. So that U of T probably really does play an important role. However, intrinsic processes still could be taking place, and they may be responsible for the transitions between states. So the bottom line here, the reason I went into all this science, is that if you want to build a nice brain-controlled interface, the more you understand about the way these neurons really operate in populations, the better you're going to do, the better engineered device you'll have. All right, that was my take-home message. This was work that was done in, over a number of years at 3 different places. In particular, Hongwei Mao, who's the current postdoc in the lab, has done a lion's share of the most recent work. Thank you very much.
Thank you, Professor Schwartz. That's absolutely fascinating. It's absolutely fascinating. And I see that you are in your, what you showed towards the end is challenging some of the ideas that exist in the field. And I'm sure there are a lot of questions that people have. I'll invite first the panelists if you have any questions, and then we'll move on to the questions from the audience.
I saw one that just popped up, and they were asking about which decoding algorithm was used in the composite video. That was basically population vectors, or more precisely, it was what we call optimal linear estimation, which is sort of an optimized population vector algorithm.
Thank you. Professor Chaudhary, Professor Vodera, Dr. Tuteja, any questions?
It's just I have one question. So I'm audible?
Yeah.
Yeah. So in terms of these oscillatory movements, if there is a repetition, or if the robo is trained to do a task, a repeated task, is there any change in these population dynamics? That means,
so, I mean, what we see is during, what I showed you is in terms of dynamics, the state of the system seems to change very quickly. So the typical arm movement lasts about 400 milliseconds. So during that 400 milliseconds, we have these 3 discrete states. And it looks like the changes between states are very abrupt. So it's almost like these step function changes. And so that suggests, in terms of a dynamic system, that there is state changes that occur rapidly and consistently. And that's the part that I think is super fascinating right now. So we don't understand how that takes place. That makes a complicated system extremely complex. And so we really need to think of, I think the overall problem needs to be thought of separately, differently, and we need a whole new way of analyzing our data. To me, as a scientist, this is super exciting. I like to find new things to discover, right? For engineers, this poses a new problem to be solved. And I think we need a new analytical tools. And by the way, you can generate the same kind of behavior in artificial neural networks. This is work that's been done, it demonstrated that you can get these state changes in artificial neural networks, in the same way. So what we're really dealing with is a complex system. It's nonlinear in certain ways. It's difficult to predict, and you can't use conventional analytical tools to try to understand what's going on.
Thanks for this nice answer. I think it was a very exciting talk. In fact, I have seen some of these work and some of these papers, and this is really an experience of looking at the way it has been framed, modeled, and actually demonstration has been built up. It was a fantastic talk. Thank you again, Professor Schwartz.
Yeah, thank you.
Professor, any other questions? We'll move on to probably Professor Richa then, so that we can take audience questions and their questions.
Thank you, Professor Jain. Professor Schwartz, thank you so much for a wonderful talk. I mean, this is a new area for me, but I got to learn a lot. I work in the area of trustable AI, explainable AI, and I was just wondering, do you envision such problems, the standard problems that we are talking about in the areas of explainability today, explainable AI? Do you envision those kinds of problems occurring in this?
I didn't quite understand the end. Could you repeat that again, please?
So, if you have followed the literature of explainable AI, we are having problems with respect to bias in the data, right? Trustability of the data, trustability of the models, bias of the models. They are more designed or tailored towards specific data sets or to a specific category of the population, depending on where the data was coming from. Do you think these kinds of problems are valid to what you are doing also, or not really?
Yeah, I think you bring up a very important point and something that's very close to my heart. It's something that I've been worried about for a number of years now. You know, as neuroscientists, we like to think of the way things work in very discrete ways. So we apply engineering principles to try to understand the system. And we think that the brain is composed of modules. And so we put each module in a little box, and that there are discrete inputs and outputs to each of those boxes, and each box has a specific transfer function. And that if we just understood all the pathways to these boxes, that we could explain function. And, you know, that's an underlying sort of almost social phenomena. And there's a certain set of assumptions and, as you said, biases to the way we look at the system. And I think as we start to think of the system more realistically and really come to grips with what our initial assumptions are and what it is we do and don't know, that we realize that we need to think about mechanism and function and description in a very different way than what we've been doing in the past.
That is very true. That is very true. Thank you for the response, Dr. Schwartz. Professor Jain, over to you.
Yeah, Professor Mayank.
So thanks, Professor Schwartz, for a very nice talk. In this whole process, see, means, again, I'm coming from a computer vision research community. So generally, we look at when we perform any vision-based task in like a neuroscience experiment and all. Many times we look at fMRI responses. What is your opinion with respect to the research you are conducting and an understanding the neural responses using fMRI? What is the future over there with respect to understanding these signals better and better? Probably via fMRI or maybe via EEG.
So I think, to me, both of those techniques are difficult to apply with these kind of analyses that we're after, where we're really interested in the sort of small time scale relationships, correlations between neurons. And it's not necessarily that these neurons are talking with each other directly. These correlational patterns could represent, you know, common drive to these large populations. And depending on the time scale of these relationships, you can't see these with fMRI. Both the spatial and temporal resolution isn't good enough. And with EEG, you can't dissociate individual neuron action with EEG. It's impossible. So these kind of relationships are, you know, in this case, bound by the methodology. You won't be able to find these. Now, in terms of state changes, if there are widespread changes in state that are somehow propagated through a population, and you can see a signature of these state changes, then, you know, and they occur on the order of 100 milliseconds, then maybe you can see this, some sign of them in the sort of gross recordings. But you won't be able to understand the details. You might see an overall signature of it, but you won't see the details. At least that's my opinion.
Sure. Thanks. Thank you very much. Professor Jain, back to you.
Dr. Sankalp.
Thank you, Professor Schwartz, for this very engaging presentation. I am from the School of Management, and a few years back, in 2018, there was a paper in one of our marquee journals in our area, Strategic Management Journal, about the gestures that Steve Jobs used to make while he used to make those iconic presentations which are so famous, when he used to introduce an iPhone and so on. And it appears that Apple has actually now figured out that engaging presentations not only are about useful slides, but also about the gestures that the presenter makes. And they are indeed training some of their senior functionaries around that. I was wondering if the model can capture some of the non-goal specific gestures that we make, like moving our hands when we talk, and similar such gestures.
That's a fascinating and interesting question, and also something that's very close to my heart. I was a student of psychophysics, these relationships of the way we move. So if you just, as you said, you look at things like gestures or any kind of movement, drawing movements, which I studied extensively, there's a lot of coordination between the joints. And it's been known for 100 years that these patterns, these coordinated patterns, are subject specific. So for instance, if you see someone in your family walking down the street quite a distance away, before you recognize any features, you'll see that there's a characteristic way that they walk, and you can recognize them by the way they walk. And just like you said, we all have our distinctive gestures. We communicate a lot of these actions and emotions through our gestures. So to cut to the chase, my goal is to decode the information with a high enough fidelity that we can capture those things. And in fact, in some of our scientific work, we've shown that we can do that. For instance, when you draw, there's a very concrete psychophysical relation between curvature and speed. Okay, it's called the 2/3 power law. It's developmental. Kids below the age of 12 don't have it. After the age of 12, people have it. It's one of those things that we use to recognize natural movement. We call it a law. So we're able to recover that from our population activity and show that in these drawing movements, that psychophysical rule was indeed obeyed. And more practically, when you watch Jan do these coordinated movements, you can see how gentle she is, right? She places these blocks in a very gentle manner. When you see our other subject, Nathan, do it, Nathan is a video game enthusiast, and he's trying to beat the clock. So he literally takes these objects and more or less throws them to a place so he can, get the shortest possible time that he wants. So yes, I think that we're very capable of decoding these sort of nonverbal, intuitive, natural features of movement in our brain activity that we record.
Okay.
Thank you. Thank you, sir.
Before I invite Dr. Rajendra or Dr. Manish or Dr. Ranju for any question, there's one question that came up from Pallavi, I think, but I am going to paraphrase it for you. So this is about movement generation. And you briefly talked about sensory inputs from the pressure sensor on the tips of the robot. But how about other sensory, primarily sensory brain-computer interface? Where are we there now?
Can you just repeat that one more time, please?
Yeah, so essentially the question is, can we actually see through a brain-computer interface or something like that? That has been a challenge for many, many decades.
I still don't, can we, I didn't quite understand it. Sorry.
So the question really is, we are getting a motor output from the motor cortex. Can
Right.
we give input to the visual cortex
Oh,
to be able to
okay,
see?
right, yeah. So there are a lot of people working on visual prosthetics, and there's a lot of technology that's been developed. Basically, the way it works, again, this is where the science and engineering differ. So the idea is that the retina and the visual system works on a pixel-based raster system, right? So the idea is that if you activate a certain spot on the retina, that activates a pixel. You put enough of these pixels together, you get an image. And so what they're doing is they're stimulating discretely, these different spots on the retina, or now even in visual cortex. And what they're generating are what's called phosphenes, little bright spots of light. And the idea is if you can put enough of these spots together, that you can see form and actually try to recreate the world. So you take a camera, feed it into a processor. The processor tells you which spots to stimulate electrically, and if you put enough of these together, you get some kind of crude image. That's the state of the art. Unfortunately, that's not the way the visual system really works. The visual system is dynamic. It relies on timing. It relies on all sorts of functions between different kinds of neurons, and it relies on speed. So the movement of the visual scene, the movement of the eyes, play key roles. So right now, the current solutions, even though they might be elegant in some of the technology they use for stimulating, really don't take into account the basic science of the visual system. And that's sort of my beef with what's going on in that arena.
Yeah, okay, thanks. So that's we need to understand and probably replicate how brain works more than what we are doing. That's what I gather, yeah. So I'll take a couple of questions again from audience. One is from Lokesh. What it says is that Neuralink company, of course, Elon Musk, whatever he touches makes it more famous than our scientists can achieve. So he just wants to know, is what he is saying as his Neuralink, is it mainly derived from early pioneers like you? I mean, I would answer it, yes, of course, this is a 2 decades old technology, but would you like to have any take on what's going on in the common press through Elon Musk company?
Yeah, so, again, I only know what I read in the newspaper. I don't really have a lot of inside information. It looks to me like the technology, the electrodes and the electronic circuits and the telemetry is, very nice, very outstanding. The packaging, and, he spent $150 million so far in developing the technology. But again, it used to be on their webpage, it says, we want, if you're interested in working at Neuralink, no experience in neuroscience required. As long as you're an engineer, come and work for us, right? And so it's almost an anti-science point of view. They think, maybe I call it engineering arrogance. Again, you might have the greatest device in the world, but if you don't understand how to use it, the utility is going to be limited.
So question is from Sumit. Actually, I was also thinking along the same line. How do we bring it to day-to-day life, what you described, and what's the limitations for that?
So again, this is something that is very close to my heart. What we've shown you so far are just laboratory demonstrations. The subjects can't really take this technology home and use it. One of the reasons is they've got these pedestals, these connectors that are screwed into their skull. sticking through their skin. And we need those to connect to our amplifiers. And they can't, that's not reasonable. You know, they get infected, they can break off. So you can't really use those in a robust way. So we need to develop telemetry. We need to have, you know, these require electronics and a certain amount of electronics and wiring inside the brain. The brain is a harsh environment. It's heated, pressurized saline. It's corrosive. So it's not, you need to have special kinds of insulation. You need hermetic sealing. And we don't have that yet. And our electrodes are fairly crude. So there's a lot of work on the engineering side that's taking place. It's going to help that. But there isn't a lot of money to make that happen. So, you know, if you try to get a grant from the government, you know, it's tough to do that. Companies aren't really interested. So we kind of have this gap between what we could be doing and what we're really doing. And that makes it frustrating. But it's all, you know, it could all be done in 3 to 4 years with a concerted effort to make it happen. So I think eventually it will. It just hasn't happened yet.
So is computational capacity is going to be one of the limiting factors which say a person can carry around or be available or something like that, or it doesn't really require very high capacity computational capacity?
You know, if we could telemetrize the signals out of the brain and just have a laptop or something, or even a small phone, you know, processor in your phone would be plenty powerful enough to do all the kind of decoding that we need to do.
Yeah, yeah. I think one clarification somebody wants to know is it a single neuron recording or multi-neuron recording? This is from Praveen Chopra.
Okay, so we try to record cleanly single action potentials from single neurons. A lot of the people in the field feel that it's not so important to be very critical about whether or not you're recording signals from one or several neurons. In the decoding algorithms that they've been using, it doesn't seem to make much of a difference. They might be correct, but I still, coming from a background where isolating single action potentials is really important, I still do that. Maybe I'm just old-fashioned, but that's what I do.
Yeah, so there is another question, you know, this is again from Praveen Chopra, that it's my understanding that these neurons, that is the relationship between firing might be non-linear. I think you've tried to address it already. Why are we using linear algorithm like PCA?
You know, one of the problems that we found is that I showed you some very simple equations, linear equations, and they account for a certain amount of the variability. But I also briefly mentioned the idea that these neurons also encode speed. And if you add these speed terms to the equation, you find that some of them, even though you add them as additive terms, when you go to decoding, there's an offset that makes the decoder nonlinear. And that really is responsible for some of the errors we see in movement. So we've come up with a hybrid system where we use both linear and machine learned nonlinear decoding to make better decoders.
And this is another, I'm paraphrasing a question. What's the difference between individuals in terms of neuronal firing, or, you know, how information is coded?
So what's really interesting is this idea of cosine tuning is very robust. So you see it, you know, everybody has it. Any mammal has it. Different, you know, grasshoppers have it. Different parts of your nervous system. You can find cosine tuning to movement throughout the entire nervous system, including sensory cortex, you know, different parts of visual cortex, superior colliculus, even primary afferents, peripheral afferents are cosine tuned for direction. So it seems to be a very robust phenomenon.
Dr. Rajendra from the panel, do you have any question?
Yeah, so my question is like how to decide the electrode density in the microelectrode array.
So, depending on the type of electrode you have, if you put in one of these pin cushions, you have to try to, if you put the needles too close together, then you're just going to press the cortex and destroy it. You need to have it so that, in theory, you sneak these electrodes in without compressing too much of the cortex. Again, people have found now that the smaller the needle size, if you get down below 5 or 7 microns in diameter, that there's basically no damage to the cortex. It's very difficult at this point to make electrodes that small. Other people are using flexible electrodes so that they're small and they're flexible. The other idea is to have multiple recording sites on a single shank. People have been working on that for years. So there's a lot of possible solutions to that. I don't think there's any clear formula for what works best at this point. It's still a certain amount of conjecture, but it seems like as technology increases, we're getting better and better electrodes, both in their ability to record and in their minimizing damage to the cortex.
Thank you, sir. Yeah, thank you.
Yeah. To what extent patient is training his or her neurons while learning to move the robotic arm? In terms of, is there any feedback which is changing the tuning of the neuron? Do you see that over a period of time?
Sure. What we showed for a very long period of time, one of our first reports, is that what happens, you can think that we have this decoding algorithm, which is an approximation to the way neurons normally fire, okay? But in this task, there's a behavioral outcome, and the monkey gets a reward for being successful and moving to the target using its brain activity. And so you can see that there's an advantage of the monkey obeying our model so that the monkey can have a higher success rate. And what happens over days and months is that you see these neurons fitting the cosine tuning model better and better. And there's a direct correspondence between how well these neurons fit the cosine model and the success rate of the monkey. So there seems to be two things going on. We try to build a model that mimics the activity, and the monkey in turn tries to learn our model. So we call that co-adaptation.
Yeah, thanks. Dr. Manish, any questions?
Yeah, so thank you, Professor Schwartz, for a very engaging talk. I noticed that you had actually used some very basic techniques like linear regression and PCA. So like moving forward, and as you mentioned that, use of nonlinear techniques like, deep learning or machine learning is possible. So in that regards, do you think the requirement of a lot of data and lots of training, would that be a barrier, or you think that would not be a challenge in applying these, nonlinear machine learning models?
Well, I think there's kind of a yin and a yang to that. Of course, large amounts of data are always advantageous for a number of reasons. But on the other hand, if you have some idea of underlying principles, you don't need brute force machine learning training. And so we try to combine two of these. We have a basic idea of the general form of the model that we're after. And right now we're using, machine learning to solve some of these nonlinear equations for us to, so we seed it with these equations and then let the model, the network, artificial neural network, solve it for us. Of course, you could just throw tons of data into it and then have the algorithm come up with some kind of output. The trouble with that, I always look at it, is that these networks tend to solve things in terms of categories. So you have sort of discrete outputs. and you're not guaranteed any kind of continuity between states that these things find. Usually, there is some, but you don't know, for instance, that, in these categories that are deciphered of the network, whether you're going to be able to interpolate and extrapolate the output. So there's this, in my mind, a certain kind of problem. In other words, if your network is trained, it might be too crispy, so that if you have, an input that it doesn't fit in, into some sort of classified output, the overfit model is going to fail. I don't know if I explained that very well, but
Okay, yeah,
that seems
thank
to
you.
be what we found.
Sure, sure. Thank you. Thanks a lot.
Okay, thank you, Professor Schwartz. That was very engaging talk and spending time for question and answers. I'm sure all of us will have a lot more questions that we'll think of it further and discuss with you, maybe offline. I'd like to invite Dr. Sankalp now for the closing.
Yes, thank you, Dr. Jain. I would like to invite Dr. Tuteja to share something from our city for Professor Schwartz. Dr. Tuteja, please.
Thank you, Dr. Sankalp. It was excellent presentation by Professor Andrew Schwartz. I'm sure we all enjoyed his talk and also benefited as well. On behalf of the School of Artificial Intelligence and Data Science of Indian Institute of Technology, Jodhpur, and also on behalf of Jodhpur City Knowledge Innovation Foundation, I would like to present a small memento to Professor Andrew Schwartz. The memento is locally produced in Jodhpur by traditional artisans using local available products. I would like to mention here that Jodhpur City Knowledge and Innovation Foundation has begun working with these 5 artisans to understand their ecosystem and support them for their livelihood, and at the same time preserving their art forms and values. Jodhpur is an institutional hub at the edge of Great Thar Desert. Jodhpur is second largest city of Rajasthan province, which is known for its historical hill forts and palaces, and it is also hub for gemstone, carpet, painting, spices, fabrics, and of course, known for beautiful camels. Thank you very much.
Thank you.
Sir, in some time, we will courier this to you, and it will reach you hopefully safely. We'll pack it well. I would now invite Dr. Ranju Mohan to deliver the vote of thanks.
Yeah, thank you, Dr. Sankar. So good evening, everyone. I'm here to give the vote of thanks for today's talk as part of our webinar series, Thar Talks AI and Beyond. We heard an excellent talk today by Professor Andrew B. Schwartz on high-performance neuroprosthetic, where he showed multi-dimensional models relating arm movement and neural activity. He demonstrated its application on animals and humans. Indeed, an inspiring work. I thank Professor Schwartz for this wonderful and informative session, and I'm sure that the findings and opinions shared by him will definitely help researchers and scientists in this area for the future research and studies. Next, I would like to thank Professor Shantanu Chaudhary, Head School of Artificial Intelligence and Data Science, for this idea of such an informative webinar series and for his continuous support and encouragement in this regard. I would like to thank CEO, Jodhpur City Knowledge Innovation Foundation, Dr. G.S. Tareja, and the entire team for the active collaboration for opening the series. Next, I would like to thank Professor Neeraj Jain for introducing such a wonderful speaker, and Dr. Rajendra, Dr. Manish, and Dr. Sankalp for the sincere effort for organizing the series. I would also like to thank Professor Mayang and Professor Richa who have been shown the support throughout the session. Finally, I would like to extend my thanks to all invited guests and other attendees for their presence throughout the session. This was the inaugural talk of the series. Next talk is on 26th May. And we will meet you all on that day. Thank you all for joining.
I think again, let us thank Professor Schwartz. It was a fantastic experience. Thank you. Good night.
Thank you. Right.
Thank you. Thanks, Andy.
Bye.
Thank you, everyone.
Thank you, Professor Schwartz.
Thank you. Good night. Bye.
Right.
Okay, Manish, बंद कर.
Rajan.
Okay, fine. Yeah.
So I think we can end this. I hope the recording would be available to us soon.
Yeah.
So we can end this. Yeah. Okay.
Thank you. Thank you, Sanjay.